In the prior sections, we saw chromosomal mutations like insertions and deletions, but now we're looking at gene mutations. Gene mutations occur during replication, and they may or may not affect the production or function of the protein for which the gene codes. There are several different types of mutation. A point mutation is the substitution, addition, or removal of a single nucleotide. So in a substitution mutation, one nucleotide replaces another. The new codon may or may not signal the insertion of the wrong amino acid. Sometimes the wrong amino acid does not affect the protein. We call that a silent mutation, mainly because of wobble. So if it's just the last letter that changes, it may not cause any big effect. So a silent mutation has no effect on amino acid sequence. A missense mutation is going to change from one amino acid to another. So instead of producing GLY, I produce SER. Nonsense mutations instead turn a codon, instead of producing an amino acid, it produces a stop, and that is going to cause some major problems for the protein itself. The deletion or addition of a nucleotide causes all subsequent codons, all downstream codons, to be misread, so they're likely to have very disastrous effects on the protein's function. That's called a frame shift mutation because it shifts the reading frame, or which codons are which. Insertions, of course, add bases, and deletions lose bases. Now, if it's an insertion or deletion of 3, it might be a minor effect, because if you lose a whole codon, the frame doesn't shift. So the worst possible frameshift mutation would be something in multiples of 2, because that's going to really throw off what the amino acids are. Because if it's only 1, wobble could make some of those changes silent, and it won't be a big deal. Now, translocation mutations are what we saw with chromosomes, large chunks of DNA swapped between non-homologous chromosomes, which can also cause some major changes. But the two most common type mutations you'll see with gene mutations are point mutations and frame shift mutations. We know that if the mutant cell is a body cell, a somatic cell, the daughter cells can be altered by the altered DNA, but the mutation will not be passed on to the offspring. If the mutant cell is a gametic cell or a sex cell, the altered DNA will be transmitted to the embryo and may be passed to subsequent generations. Gamete cell mutations result in genetic disorders, whereas a body cell mutation could be something like cancer, and you're not going to pass that on to your offspring. Outside of mutations, there's this big field of biotechnology that we use to understand our DNA, understand diseases, find ways to improve our lives. Biotechnology utilizes biological processes, organisms, cells, or cellular components to develop new technologies. New tools and products are developed. It's useful in research, agriculture, industry, medicine, wide variety of purposes. The oldest form of biotechnology or genetic engineering is just selective breeding. Basically, humans control what mates with what, and that is going to be an artificial selection. So we know about natural selection. Artificial selection is much faster because you're not leaving it up to random chance. So, for example, we took Zea mays from the ancestral teosinte at the top right over here, and we eventually, through selective breeding, produced our modern corns. On the left, we have the wild mustard plant that we bred into cabbage and cauliflower and kale and Brussels sprouts and broccoli. So selective breeding is a very fast form of natural selection, but it's because humans do it, it's artificial selection, and it is the oldest form of genetic engineering. Now, what we really think of as genetic engineering is a bit more complicated. Genetic engineered products currently on the market include human insulin produced by genetically modified bacteria, plants with resistance to certain insects, plants that can tolerate certain herbicides. Basically, you take genes from one organism and you put them in another, and because the genetic code is universal, it works. Any organism that we create through genetic engineering is called a genetically modified organism, or a GMO. But just because something is genetically modified does not mean it's necessarily a bad thing or is going to cause problems. Some of that hype is a bit overrated. Another part of biotechnology is the sequencing of DNA, because if we can sequence DNA, we can understand what genes do, we can use those genes in genetic engineering. There's two major ways that DNA is sequenced. You have the old-fashioned map-based method, which is we've used for many, many years. You cut that DNA into a lot of small segments, you line them up, and you cut and clone and sequence. It's a slow process, but it's a very effective process and doesn't lead to a lot of errors. A newer method, which was created by Vintner and others, was the shotgun method. You basically cut the DNA into tiny, tiny fragments. The fragments are so small, they're very easy to sequence, and then you arrange them based off of overlapping sequences. There is the potential for error here, because remember, we're looking at 3 billion base pairs, but it is extremely fast and extremely cheap. Restriction enzymes are enzymes that cut DNA at very particular sequence. These evolved to basically fill in the role of an immune system in a bacteria, and it's going to help protect it from any damage. Restriction enzymes, each of them cut at a different place, and we can use them to cut DNA in very controlled fashions, and allows us to also insert genes for the coding and cloning of genes. When you cut up DNA using restriction enzymes, you get very particular sequences. Because each restriction enzyme cuts at a certain place, you can predict what sequences will be common. If you put that cut DNA into a agar gel and you run it through gel electrophoresis, what you see are bands of color, and every band is going to correspond to sections of DNA. DNA is negatively charged, so it moves towards the positive end. The smaller factors are going to go through the gel faster because they're smaller, they can get through the holes in the gel pretty quick. And the longer molecules are going to stay up towards the top because they're having problems getting through. So if I use a DNA ladder or some source of comparison, I can compare the gene bands that I see to test for things like heredity and paternity and diseases and a wide variety of other factors. So once DNA is cut and it's run through gel electrophoresis, you can compare the bands that are produced with a standard or with other samples to test for relatedness or heredity or to identify a person, because the specific patterns of bands produced by electrophoresis create a DNA fingerprint. Those pieces form what's called the RFLP, or RFLP, Restriction Fragment Length Polymorphism, and everybody's RFLP patterns are going to be different. But they're going to be closer between you and a relative. The first ever case that this was used forensically is shown below, where the semen sample from the rapist was matched against a blood sample from the suspect to determine that the rapist was in fact guilty. Taking pieces of restriction enzymes and all these things together, we can use the natural reproductive capability of bacteria and plasmids to clone and copy genes that we want. So recombinant DNA is when you take a DNA sequence and you insert something else into it to make a new DNA strand. So bacteria have these little plasmids, and by cutting a plasmid with the restriction enzyme and by cutting the gene that I want to copy with the same restriction enzyme, we can make it so that the gene and the plasmid match up and they join together. We then throw in some ligase and they glue together to form one cohesive plasmid. Then you let the bacteria copy, and then you extract what you wanted. This is how, for example, we copy human insulin. It would be very hard to grow enough insulin to keep everyone alive based off of human sources. So we use bacteria to grow that insulin for us many, many times over more than a human could produce. When you think about biotechnology, the thing that most often pops into your mind is cloning. There are a lot of types of cloning. There's the natural cloning that organisms do through asexual reproduction, which is nothing really new. Molecular cloning, bacterial plasmid vectors could be considered molecular cloning. There's also the more advanced polymerase chain reaction, which allows you to make thousands to millions of copies of a segment of DNA or a gene to study, which could be very useful. But cell cloning is what people think about most of the time when they think about cloning. To do that, you take the nucleus out of a donated egg, put in the nucleus of the organism that you want to clone, and allow that egg to grow normally in a womb. It's very labor intensive and it's poorly understood. For example, Dolly the sheep was born after 277 eggs were used to create 29 viable embryos, and only 3 of those embryos survived until birth, and only Dolly survived to adulthood. Other examples are CC the cat, which, funnily enough, is CC stood for carbon copy. And that cat was a clone, but it didn't look like what it was copied from because of X inactivation and a few other factors. So cloning, it's really poorly understood, it's not very well done, and it can't be automated, so it's very labor intensive. Plus, you can't remove the mitochondria and everything from that cell, you're only removing the nucleus. So some genetic components will be unchanged, so making a true clone is actually extremely hard.